Without further ado, we shall now move on to Oksana Beskranava, and representing Dyne Therapeutics. I shall hand over to you, Oksana.
All right. Good afternoon, everybody. And thank you very much for this opportunity to be here with you today. And I'm very happy to be able to share with you our new and exciting data that demonstrate power of our FORCE platform to enable muscle-specific delivery of therapeutic oligonucleotides for the treatment of Duchenne muscular dystrophy. This is my forward-looking statement. I will be making certain forward-looking statements that involve some risks and uncertainties. I would like to start with the introduction of our FORCE platform. I would like to review its design, describe components of the platform, and explain how we design the FORCE platform to enable muscle-targeted delivery of oligonucleotides. And then I will discuss the application of our FORCE platform for the treatment of Duchenne muscular dystrophy, and specifically we'll focus on preclinical efficacy in a well-known MDX mouse model and ability of FORCE to achieve robust and durable expression of dystrophin in skeletal and cardiac muscle. And then I will share with you our data that supports specifically our path to clinic, focusing on safety and exon skipping of our clinical candidate named Dyne 251, and data will be presented from non-human primates. Our FORCE platform contains three essential components. The first important one is the antibody, or specifically it is a Fab fragment, which is designed to bind transferrin receptor 1, or TFR1, to facilitate targeted delivery to muscle, a clinically validated linker, and a payload. Importantly, payload is designed rationally to address the genetic basis of the disease. Today we will be talking about oligonucleotide payloads, but other type of payloads can also be used if desired. Our FORCE platform allows sufficient flexibility to deploy these different types of therapeutic payloads. And as I mentioned, they are designed to match target biology that is different in each disease indication. For example, we would choose a single-stranded ASO payload in order to enable targeting nucleus, or siRNA if we are looking for cytoplasmic targets to modulate. For our Duchenne muscular dystrophy program, we utilize ASO called PMO, which is effective in driving nuclear exon skipping. And again, we would leverage siRNA if we desire to modulate cytoplasmic targets. So our FORCE platform is specifically designed to enhance muscle delivery by leveraging TFR1 expression on skeletal, cardiac, and smooth muscle. And we aim to deliver redosable, titratable, and durable therapy. It is designed to target the genetic basis of disease, and we rationally select our payloads to match target biology with enhanced tolerability. And finally, I'd like to mention that we use the same Fab and the same linker across all disease indications. And that allows us to reduce our development and manufacturing costs, which is not trivial. Duchenne muscular dystrophy, as you know, results from genetic mutation in DMD gene that encodes dystrophin, a protein which is essential for a normal muscle function. And in people with Duchenne muscular dystrophy, dystrophin is absent or greatly reduced, which causes permanent damage to muscle cell, leading to muscle weakness, progressive loss of function, cardiac and respiratory failure. Available exon skipping therapies have demonstrated increased, but yet limited dystrophin production. And so we set out a goal for our DMD program to achieve robust exon skipping, to significantly increase dystrophin production with also less frequent dosing. And we aim to potentially stop or even reverse the disease. Now we've generated a very exciting set of data in the program, starting with our lead program in exon 51. This schematic just depicts a mechanistic approach aimed at exon skipping. In this case, we're looking for the treatment of exon 51 skipping amenable DMD patients. So dystrophin gene is the largest human gene with 79 exons, which are spliced together for a functional full-length protein. In Duchenne muscular dystrophy, for example, with deletion of exons 49, 50, the reading frame is lost and no dystrophin is produced. With a therapeutic oligo that causes exon 51 skipping, reading frame is restored and shortened, but functional dystrophin protein can be expressed. Let's review our preclinical data that show how we use our FORCE platform for exon skipping therapy in Duchenne muscular dystrophy. We have started these studies by using established MDX mouse model. Just a reminder that it has a mutation in exon 23. The idea here is to demonstrate proof of concept for the approach for the FORCE platform, because we will be using a surrogate Fab and exon 21 skipping oligonucleotide. So our initial study were short-term, only 2-week in duration. And we are looking here at a single dose of delivered IV, single dose of 10, 20, or 30 mg per kg. You are looking at Western blot at left-hand side, 2 weeks post-injection, and on the right-hand side, you're looking at measurements, suggesting that with this type of schedule, we can easily restore up to 10% dystrophin with no associated dose-limiting toxicities. In comparison with naked PMO, in this setting, one can only produce about 1% of dystrophin. With the same type of approach, we conducted functional studies in MTX mice, running wheel and hind limb fatigue challenge, which is on the right. On the left-hand side, you're looking in 12-hour longitudinal study of mice that are performing on the running wheel. The y-axis is the distance that mice are able to travel. X-axis is time of the day. And if you look at between the two vertical dashed line, which depicts a dark period when mice are most active, you can see that gray circles depict wild-type mice, and black squares show MDX mice, and you can see the difference in their activity. And when you compare performance of MDX mice treated with FORCE platform, shown in blue triangles, we are showing that the function is restored almost to the wild-type levels. On the right-hand side, you see results from the hind limb fatigue challenge. In this case, mice are required to stand for about 10 minutes on their hind limbs, and then we measure loss of range of motion. So obviously, gray bar showing wild-type mice show the least amount of motion loss, 20% of range of motion. You can see in black MDX mice, which lose about 70% of motion. And then red bar shows that treatment with naked oligo do not provide benefit to MDX mice. But blue bar shows that it's non-statistically different from the wild-type range of motion. So this was, again, relatively short-term studies, and in order to better understand the dynamics of the FORCE platform, we set out the study where we are looking at kinetic pharmacodynamic of FORCE on exon skipping and dystrophin expression, dosing using, again, a single dose of either 10 or 30 mg per kg that is administered once. And then we're collecting tissue at day 3, 1 week, 4, 8, or 12 weeks, as indicated with these red squares. Endpoints we collect, we measure tissue concentration ASO, we measure exon skipping and dystrophin protein by either Western blot or immunofluorescence. And we are looking at 3 tissue: quadriceps, diaphragm, and heart. So this slide captures the results of the study with a dose of 30 mg per kg of FORCE. So the top panel with blue lines show the analysis of skipping. The green lines below show dystrophin protein expression by Western blot. So you can see that in all three tissues that we analyzed, we achieve robust and durable skipping as well as dystrophin expression. For example, if we look at quad in the left panel, we see that dystrophin is detected up to 12 weeks post-dose. At 4 weeks, we detect about 46% of dystrophin in quad, 90% in diaphragm, and 78% in the heart. So let's take a look closer in each tissue. So in the next few slides, I will review data in quadriceps. So here on the left-hand side, you see dose-dependent measurement of exon skipping that peaks at about week 1, both with 10 and 30 mg per kg, and is detectable for up to 12 weeks with a higher dose, with 30 mg per kg. The right panel shows dose-dependent dystrophin production that is highest at week 4, but also lasts up to 12 weeks with the highest dose. Now here we are looking at 8-week time point. With the left panel, it's a visual dystrophin expression by Western blot. And you can see clearly a nice dystrophin band in each individual animal with very low level of heterogeneity. In the right-hand side, you see dystrophin localization, again, at 8 weeks of age to the sarcolemma. The staining is uniform with about 83% of positive fiber. And I would like to also draw attention to the laminin staining that's shown here in red that helps us visualize tissue architecture. And so what you can see is that treated quad muscle with force shows tissue architecture that is significantly improved, unlike what you see when you look at MDX tissue. And finally, I would like to take you to take a look at all this data put together in the dynamic, showing dystrophin membrane localization up to 12 weeks. So from this slide, you can appreciate how effectively force is able to re-express dystrophin at the muscle membrane with 68 to 83% of positive fiber at week 4 and 8. And importantly, we still detect membrane-associated dystrophin up to 12 weeks, which is extremely exciting finding for our fourth platform. Now let's take a look at diaphragm data. Again, here we achieve effective dose-dependent exon skipping. And again, we see the exon skipping peak at about 1 week. And we observe up to 90% dystrophin expression level by Western blot. At highest is now at 4 weeks post-dose. The next slide shows detection of dystrophin by Western blot on the left-hand side and localization in the diaphragm on the right-hand side. Again, we observe a very high number, 80% of dystrophin-positive fibers in the diaphragm. And as you know, this is a very important tissue for us to target for the therapeutic benefit in Duchenne muscular dystrophy. This slide now captures our data in the heart. On the left, you see exon skipping that is achieved in the heart that translates into robust dystrophin expression by up to 78% at week 4. And we can still see a detectable level of dystrophin at week 8 in the heart. And again, we see the dystrophin protein here on the left-hand side with Western blot, and the right panel shows localization of dystrophin to cardiac muscle membrane. At 4 weeks, you're looking at the data at 4 weeks, 78% of dystrophin expression and 80% of dystrophin positive fiber. So this is very exciting data that allows us now to capture all of this together in a single slide that you can see side by side in all this tissue that we just went through. And what we are looking at is robust and durable dystrophin expression with proper membrane localization in both skeleton and cardiac muscle. To put this data in a visual summary of our actual modeling of pharmacokinetic pharmacodynamic, we generated this analysis with 10 mg per kg cohort, which is on the left panel, and 30 mg per kg cohort on the right panel. And this allows us to take a look at pharmacokinetic pharmacodynamic profile that allows us to deliver substantial and durable dystrophin expression with single dose. So if you look at black line, we are looking at tissue ASO concentration. Blue line shows exon skipping, and green line is dystrophin expression. Importantly, we achieve peak ASO concentration relatively fast, in less than a week. And then that leads to the peak of skipping around 1 week post-dose, and dystrophin expression that peaks at about 4 weeks post-dose. So these data uniquely describe how FORCE platform works and PK/PD relationship that is associated with the way we deliver. And we deliver oligonucleotides via transferrin 1 receptor. And I would like to emphasize that we are utilizing natural cell biology to be able to deliver our oligonucleotides within the cell. So these dynamics are specific to the FORCE and could be different from other types or other modalities that can be used in order to facilitate delivery of oligos into the muscle tissue. And these data are an important step for us for human translation. Now, on our path to human translation, I would like to share new data using our clinical candidate, Dyne 251, using DMD cells and non-human primates. In DMD patient-derived cells that are amenable to exon 51 skipping therapy, we show that Dyne 251 achieves robust and dose-dependent exon skipping. And next, we tested the ability of Dyne 251 to achieve exon skipping in non-human primates in vivo. Just wanted to remind you that in NHPs, we are looking into skipping of normal dystrophin RNA, where we introduced the de novo stop codon, so we are not looking at the disease tissue. We tested different regimens. So what I would like to just show here that we used two different arms. First, green triangles depict a single IV injection, and red square shows the time of tissue collection. Each dose is a low dose of 30 mg per kg. First, we set out to analyze the arm that is only lasts for 1 month post first dose. So what I am going to show you is data cut at 1 month, where we either administered 30 mg per kg every week or every other week. And then we have a longer arm that is designed to take these animals off drug after week 4 and let them sit off drug for another month with data collection at 2 months time point. So with that schedule, here is what we observed. Interestingly, we achieved roughly similar exon skipping at 1 month with either schedule. We can do 4x30 schedule or 2x30 mg per kg. We observed the same amount of skipping. When we collected tissue a month later with no drug treatment in that time frame, we obtained profound skipping. And now we observe up to 18% of skipping in quadriceps, 52% skipping in diaphragm, and 43% skipping in heart. So these results show that we are able to achieve in higher species significant skipping of dystrophin RNA, even though we are looking at a normal production of mRNA. And especially we see a high number of skipping in heart and diaphragm, very important tissues that lead to mortality over time in patients with DMD. Our NHP toxicology results showed no dose-limiting toxicity after 5 weekly doses that we continue to raise up higher to a maximally feasible dose that we can formulate. And despite pushing the dose to maximally feasible dose and using this aggressive weekly schedule, we observed no changes in cardiac, respiratory, neurologic, or ophthalmic parameters. We have no effect on kidney or liver function or coagulation. And importantly, that no AL dose was identified as the highest dose we were able to test. Therefore, our GLP toxicology results support advancement of Dyne 251 to clinic. In summary, in MDX model of DMD, FORCE platform achieved dose-dependent, robust, and durable exon skipping and dystrophin expression, long-lasting dystrophin localization to the sarcolemma, and produced functional benefit. Dyne 251 has shown robust exon skipping in DMD patient cells, in NHP cardiac and skeletal muscles. And importantly, this data, along with a good profile in NHP toxicology, support advancement of our molecule Dyne 251 into the clinic. So with this, I would like to acknowledge our entire Dyne R&D team for their creative work that I had a pleasure to share with you today. Thank you for your attention, and I will be happy to take questions.
Thanks very much, Oksana. Very clear presentation. There is one question, and I think we'll only have time for one, I'm afraid, but a question about how your data compare to those already available about exon 51 skipping ASOs or other exon skipping ASOs.
What we can say that if you look in MDX data, we are showing a profound effect with only single dose, and we can see not only production of dystrophin, but smooth overall localization, continuous localization along the plasma membrane. I think these data are sets this type of approach quite apart in terms of the amount of dystrophin we delivered and longevity. If you look at NHP data, it is known that PMO are naked PMO produce benefit upon chronic dosing, although amount of dystrophin produced is very tiny. And what sets us apart is ability of our platform to deliver to, for example, to the heart, where naked PMO is notoriously not capable of penetrating.
Great. Thank you very much.